Hello everyone, Biochemistry web channel is here with the topic titled Amino Acids, Peptides and Proteins-II. Please visit my website biochemia.web for a more comprehensive document of the topic in PDF format. You can find the link in the description. And please don't forget to like the video before you start. I wish it will be useful for you. Let's get started. Let's look at the content of our topic, and we will start with the peptide. What is the peptide bonding and oligopolypeptides and the other titles. And then proteins, which is a big part of the topic. We will examine the structure, properties, and classification of proteins. Yes, the first title is peptide. And we'll talk about peptide bonds, oligo and polypeptides, some important ones, and then some methods to separate amino acids and the quantitative determination. Just some brief information about the method. Amino acids polymerize via acid amide bonds, which we call them peptides, from peptides and proteins. Two amino acid molecules can be covalently joined through a substitute amide bond called a peptide, and this is called a dipeptide. Peptide bond formation is an example of a condensation reaction, a common class of reactions in living cells. Under standard biochemical conditions, the equilibrium of the reaction favors amino acids over the peptide. To make the reaction more thermodynamically favorable, the carboxyl group must be chemically modified or activated so that the hydroxyl group can be more easily eliminated. Three amino acids can be joined by two peptide bonds to form a tripeptide. Similarly, four amino acids can be linked to form a tetrapeptide. When a few amino acids are joined in this way, the structure is called an oligopeptide. Peptides are concise sequences of amino acids connected through peptide bonds. A polypeptide refers to an extended, uninterrupted, and unbranched chain of peptides. It's also present in the following slides that proteins can have thousands of amino acid residues. Although the term protein and polypeptide are sometimes used interchangeably, molecules called polypeptides usually have a molecular weight below 10,000 Dalton, and those called proteins have a higher molecular weight. Proteins are specifically identified as polypeptides with a molecular mass equal to or greater than 10,000 Dalton. And oligopeptides encompass chains comprising fewer than 20 amino acids, such as penta, hexa, or decapeptides. Since the carboxyl group of one of the amino acids, as you see here, and the amino group of the another amino acid are chemically bonded during the formation of the peptide chain. This is a condensation reaction. 1 mole of water is removed. And as we know from the amino acids, and amino acids always carry an N-terminus and carboxyl terminus. Thus, at the end of the peptide formation, also you see the water here. Here is a dipeptide, or maybe tetra, penta, hexa, or in a polypeptide, there is always an N-terminal, which is a free amino group at the one end of the chain, and the carboxyl terminal, or C-terminus, C-terminal, and also for an N-terminal or N-terminus. One N-terminus and the other end of the polypeptide chain, there is an C-terminus. Like free amino acids, peptides have characteristic separation curves and a characteristic isoelectric point, which is pi at a certain pH. So they don't move at isoelectric point. And here is some important the oligo and polypeptides, and as you see the carnolin and the peptide or antirin tripeptide. the oxytocin was a protein 9, which means non, and the more ones, the polypeptide ones here, insulin glucagon. No generalizations can be made about the molecular weight of biologically active peptides and proteins in relation to their function. Naturally occurring peptides range in length from 2 to thousands of amino acid residues. Even the smallest peptides can have biologically important effects. Consider the commercially synthesized peptide aspartyl phenylalanine methyl ester, artificial sweetener better known as aspartame or Nutritive. And also you see here the glutathione, the tripeptide, which is involved in oxidoreduction reaction. It's a potent antioxidant and also the structure of some enzymes. And also, as you see, there are hormones here. Oxytocin, vasopressin, ACTH, adrenocorticotropic hormone, insulin, glucagon. These are hormones. They are oligo and polypeptides. Many small peptides in here, such as oxytocin, so exert their effects at very low concentrations. For example, a number of vertebrate hormones are small peptides. Oxytocin, for example, nanopeptide, and vasopressin, and another nanopeptide. And they are the hormones of pituitary gland. And they are secreted by the posterior pituitary gland or released by the posterior pituitary gland and stimulate some functions such as oxytocin stimulates uterine contraction. And another example can be thyrotropin releasing factor, which the TRH, which induce which is secreted by the hypothalamus and induce pituitary gland to secrete thyrotropin or the thyroid stimulating hormone. And how long are the polypeptide chains in protein? The lengths vary considerably, such as human cytochrome C has 104 amino acid residues attached to a single chain. But bovine chymotrypsinogen has 245 residues. And at the extreme end is the chitin, a component of vertebrate muscle, which has about 27,000 of amino acid residues and a molecular weight of about 3 million Daltons. And the vast majority of the naturally occurring proteins are much smaller than this and contain less than 2,000 amino acid residues. Here you see the famous insulin, a polypeptide secreted by beta cells of the islets of Langerhans of the pancreas. And it is synthesized as a pro-insulin, as you see here, forming a 21 alpha chain and 30 beta chain and 31 C peptide chain. And in the active insulin, the C peptide, which is the connecting peptide of alpha and beta chains, are not present. Is not present. And here is a difference between human and cow pig insulin. And human and pig insulin only differ in one amino acid in the beta chain at the carboxy at the C terminal. Instead of threonine, pig carry alanine, and in the cow there are three differences comparing to human insulin. Cow, similar to pig, alanine in the beta chain C terminal, and two are in the alpha chain. Instead of threonine and the isoleucine, cow carry valine. And as you may know, the insulin is so critical hormone. And I also say it's a lone ranger in the body, which regulates it, which has effect on carbohydrate metabolism as well as in the fat metabolism and protein metabolism. And it is the only hormone that has effect in decreasing blood glucose concentration. Let's look at some analytical methods or some methods we use in studying amino acids, peptides, and the proteins, which they may be quantitative and or qualitative. And as you see here, the chromatographic method based on the absorption, paper, ion exchange, thin layer, or high pressure liquid chromatography, isotope deletion method, and also there are microbiological methods. But recently, the most notable and widely used ones are actually mass spectrometry, electrophoresis, and as well as the chromatographic techniques. And what are the principles of these techniques? The chromatographic techniques based on using the adsorbents, different absorbents, and amino acids, for example, absorbed by the basic absorbents and basic ones are absorbed by the acid absorbents. And in isotope deletion method, in an unknown amino acid mixture, after adding a known amount of certain amino acid labeled with an isotope atom, it is determined to what extent the amino acid is diluted after obtaining pure amino acid from this mixture. In the microbiological method, it's an event based on the principle that certain amino acids must be present in certain amounts in the growth medium for some organism to grow. And thus to the major title of this topic, the protein structure. We will look at structure of proteins and denaturation, general characteristics, and classification. Proteins are usually made up of thousands of amino acid residues. Although there is sometimes similarity between the term protein and polypeptide, as I mentioned before, generally molecules with a molecular weight of less than 10,000 are called polypeptides, while those with a higher molecular weight are proteins. If there are n number of amino acids in the protein structure, there are n minus 1 number of peptides. What makes one protein an enzyme, another a hormone, another a structural protein, and another an antibody? How do they differ chemically? The most obvious distinctions are structural. And now it's time to look at protein structure. The structure of large molecules like proteins can be described in various levels of complexity, organized in a kind of conceptual hierarchy. 4 levels have been identified to define protein structure: primary, secondary, tertiary, and quaternary structure. What is the primary structure? It refers to the chain-like structure formed by a certain number of amino acids arranged in a certain sequence or the primary structure of all covalent bonds, the main peptide bond, linking amino acid residues in a polypeptide chain. The most important element of the primary structure is the sequence of amino acid residues. Secondary structure refers to particularly stable arrangements of amino acid residues that lead to recurrent structural patterns. Alpha helix, formed when the peptide chain is twisted into a helix around an axis. Beta sheet, a structure formed by 2 or more polypeptide chains joined together by hydrogen bonds. Also, there are other ones such as beta turn. The tertiary structure, helix structures are folded by phase to become globular and ellipsoidal. The tertiary structure describes all aspects of the three-dimensional folding of a polypeptide. The quaternary structure, when a protein has 2 or more polypeptide subunits, their arrangement in space is called a quaternary structure. And these are short definitions that I did for you. We will go a little deeper in a moment in the following slide. And here you see the hierarchy of the structure of the proteins from primary to secondary. And here you see the alpha helix and the tertiary structure in a polypeptide chain and more combined to form or assemble subunits to form the quaternary structure. The difference in primary structure can be particularly informative. Each protein has a unique number and sequence of amino acid residue. And in the given example, you see the residue, prolamin, alanine, and other ones. The protein's function depends on its amino acid sequence. The bacterium Escherichia coli produces more than 3,000 different proteins. Human has approximately 25,000 genes encoding a much larger number of proteins. In both cases, each type of protein has a unique amino acid sequence that provides a specific three-dimensional structure. This structure in turn gives it a unique function. Amino acid sequences provide important biochemical information. Knowing the amino acid sequence in a protein can provide insights into its three-dimensional structure and function, cellular location, and evolution. Much of this information is obtained by searching for similarities between a protein of interest and previously studied proteins. Thousands of sequences are known and available in databases accessible via the internet. The comparison of a newly obtained sequence against this larger bank of stored sequences often reveals relationships that are both surprising and illuminating. Secondary structure is the alpha helix. The term secondary structure refers to any selected segment of a polypeptide chain and describes the local spatial arrangement of the main chain atoms, regardless of the position of its side chains or its relationship to other segments. And there are several types of secondary structures that are particularly stable and commonly seen in proteins. The most prominent are alpha helices, as they see here, and the beta sheet or beta conformation. Another common type is beta turn. In the absence of a regular pattern, the secondary structure is sometimes referred to as an undefined or random helix. The path followed by most of the polypeptide backbone in a typical protein is not random. Rather, it is invariant and highly specific for the structure and function of the protein in question. The tertiary and quaternary structures you see here, polypeptide chain and the assembled subunit. And the overall three-dimensional arrangement of all atoms in a protein is referred to as the tertiary structure of the protein. The term secondary structure refers to the spatial arrangement of adjacent amino acid residues in a portion of a polypeptide, while tertiary structure involves longer range aspects of the amino acid sequence. Amino acids that are distant from each other in the polypeptide sequence and in different types of secondary structure can interact within the fully polluted structure of a protein. The location of folds, including turns, and the polypeptide chain and the direction and angle of these folds are determined by the number and location of specific fold-producing amino acid residues such as proline, threonine, serine, and glycine. And interacting segments of a polypeptide chain are held in their characteristic tertiary positions by various weak interactions between the segments, and sometimes by covalent bonds such as disulfide crossing. Some proteins contain two or more separate polypeptide chains or subunits, which may be the same or different. The arrangement of these protein subunits into three-dimensional complex forms the quaternary structure. As you see, a polypeptide chain, and here there are four polypeptide chains as subunits form the quaternary structure. And given these high levels of structure, it is useful to identify the main groups into which many proteins can be classified. And fibrous proteins, which have polypeptide chains, are arranged in long strands or sheets, and globular proteins, in which the polypeptide chains are folded into spheres or globules. and we will look at this classification in a moment. Another important title, Denaturation of Proteins. Protein structures have evolved to function in specific cellular environments, and conditions different from those inside the cell can cause changes in protein structure from large to small. Denaturation is the loss of three-dimensional structure in a protein molecule that causes loss of function. In short, denaturation is the disruption of the natural structure of protein. Unfolding and refolding of the protein molecule in a different way, for example, changes in the secondary and tertiary structures, change the properties of the protein molecule. It's not necessary for peptide bonds to dissolve for denaturation, although the disruption of the tertiary structure is a reversible denaturation. In cases where the secondary structure is disrupted, the resulting denaturation is irreversible. The denaturated state does not necessarily equate to a complete unfolding of the protein and randomization of the conformation. Under most conditions, denaturated proteins exist in a series of partially folded states. In other words, the protein molecule must be exposed to a number of factors in order for the native structure to be disrupted. Protein denaturation involves disruption of both secondary and tertiary structures, and denaturation reactions are typically not strong enough to break peptide bonds, and the primary structure remains the same after denaturation. And here is a reaction wave of denaturation. And as you see the normal protein and the denaturated protein, which it lost its tertiary and secondary structure. And in some cases, the native protein structure can be restored, and this is called renaturation. And denaturation causes loss of biological activity. And if there is a complete renaturation, the recovery of biological activity happens. Protein denaturation is often not reversed, and the protein native state cannot be restored. Most proteins can be denaturated by heat, which has complex effects on many weak interactions in the protein, primarily in hydrogen bonds, providing the shape of the protein, the secondary and the tertiary. If the temperature is slowly increased, the conformation of a protein usually remains intact until a sudden loss of structure occurs within a narrow temperature. And the suddenness of the change suggests that folding is a cooperative process, and loss of structure in one part of the protein destabilizes the other. Proteins can also be denaturated by extreme pH values, another pH. So we have pH, temperature, and then the pH. And certain miscible organic solvents such as alcohol or acetone, or certain solvents such as urea and guanidine hydrochloride or detergent can also cause denaturation of proteins. They change pH. And each of these denaturating agents represents a relatively mild process in the sense that the covalent bonds in the polypeptide chain are not broken. In organic solvents, urea, detergents, organic solvents, urea, and detergents act primarily by disrupting the hydrophobic interaction that form the stable core of globular protein. And urea also disrupts hydrogen. An extreme pH change the net charge on the protein, causing electrostatic repulsion and the breakdown of some hydrogen bonds. Thus, the acid-base balance of an organism is so important in terms of the native state of protein. Denaturation usually leads to protein precipitation, which is a result of protein aggregate formation by association of exposed hydrophobic surface. and the aggregates are usually highly dissolved. An example is the protein precipitate seen after boiling egg white. And as well as we shall see, more ordered aggregates are also observed in some proteins. And a good illustration, thanks to Professor Tenchov, a reversible and irreversible denaturation. He used the native state of a globule, and after denaturation, a random coil is formed. If the renaturation occurs completely, the protein can gain its native state. Otherwise, the aggregates occur, and the major cause leading to irreversibility of denaturation is the formation of aggregates from the denaturated protein molecule, so that the restoration of the original native structure is practically impossible. And protein denaturation is often irreversible. An important reason leading to the irreversibility of denaturation, as I want to mention again, formation of aggregates from denaturated protein molecule, so that the restoration of their original native structure is practically impossible. What are the general characteristics of proteins? Molecular weight. Proteins are substances with high molecular weight. Their molecular weight increases with the formation of the quaternary structure and decreases with the breakdown into monomers. Therefore, the molecular weight varies according to the conditions or between the protein molecules, such as hemoglobin, approximately 64,500 Daltons, a big protein. Protein is a larger molecular weight. What about insulin, a polypeptide, about 5,800. Precipitation of proteins. Proteins precipitate with certain acid, heavy metals, and specific antibodies. For example, mercury, lead can cause precipitation. And also, each protein precipitates with specific antibodies against its own species. And also, this is the basis of antibody-antigen reaction or defense against pathogens or foreign proteins. The molecular shape of proteins, in general, they form globular or fibrillary structures that we will classify them according to their structures. Hydration of proteins, they bind water because they carry polar group, amino, carboxyl, and some amino acid hydroxyl group, amino group, etc., which they, due to these groups, they have the ability to bind water. Viscosity of proteins, it is directly related to the shape of the molecule. If the molecule is long, viscosity is higher, and also viscosity is also higher in proteins with larger molecules. In other words, another definition. And the crystallization of proteins, enzyme proteins such as pepsin, trypsin, and urease were obtained in crystal form. And these are some basic information about the character. Classification of proteins, and there are two types that we can do. The one is structural classification, and another one is classification according to their biological roles or functional classification of proteins. And we will start with the structural class. And in this class of proteins, there are simple proteins, which they have the polypeptide chains composed only of amino acids. There are two types, globular and fibrillar proteins or fibrous proteins. There are conjugated proteins. These are proteins that give chemical substances other than amino acids when hydrolyzed, but these are the proteins contain different class of compounds, such as carbohydrates, for example. And also there are derivative proteins. And for the derivative proteins, please visit the website to download the PDF document. I will not talk about the derivative proteins here. Simple proteins, the globular ones. And the globular proteins are one of the two main protein classes. Other is fibrillar or fibrous protein. And they are named for their roughly spherical shape, and they are highly functional within organisms. They are generally soluble in water and other polar solvents, which allows them to play dynamic roles in cellular processes. And the solubility is due to the hydrophilic, which means water attracting R groups of amino acids that are usually oriented on the outside of the protein molecule. And globular proteins are divided into subgroups such as albumin, globulin, globins, and others. Let's start with the albumin. And albumins are the globular proteins having molecular weight below 100,000 Dalton. And in general, they are the lysine poor protein. And here are some examples of albumin, serum albumin, lactalbumin, and legumin. Plant source, egg, serum albumin in the blood serum, and lactalbumin in milk. And here you see the 3D shape of the serum albumin. Serum albumin is the most abundant blood plasma protein. And serum albumin serves as the carrier of fatty acids, bilirubin, ions, and drugs. Globulins are a family of globular proteins that have higher molecular weight than albumin. Here you see they have the higher molecular weight, over higher than 100,000 Dalton, and they are lysine-rich proteins. Some globulins are produced in the liver, while others are made up by the immune system. And globulins, albumin as globular proteins, or serum albumin or serum globulins, which are the alpha, beta, and gamma globulins, and the fibrinogen, which is a fibrillar protein, are the major blood proteins. Globins. Globins are a family of globular proteins which are heme-containing proteins involved in the binding and transport of gases like oxygen and carbon dioxide. And the most well-known globin is hemoglobin, found in the red blood cells of many animals, which is responsible for oxygen transport from the lungs to the tissues and the return transport of carbon dioxide from the tissues to the lungs. Myoglobin is another example for globins found in muscle tissues, where it serves as an oxygen store and facilitates the transport of oxygen within muscle. And the other example is neuroglobin, which is found in the brain and the retina, and it is thought to protect these tissues from hypoxia by delivering oxygen. Glutelins are a class of proteins found primarily in the seeds of cereal. Glutelins are storage proteins, meaning they act as a reserve of amino acids for the plant embryo used during germination and early growth. And some examples are glutenin in wheat, ordenin in barley, and orzenin in rice. Histones are highly alkaline proteins found in the nuclei of eukaryotic. Histones are highly alkaline, which they carry the basic amino acids in higher ratio, lysine, histidine, and arginine. And in the nuclei of eukaryotic cells, histones exist as a package and order DNA into structural units called nucleosomes. And they are critical for the compaction of DNA into chromatin, which allows for the efficient storage of a large amount of genetic information in the cell nucleus. Prolamins. Prolamins are a group of plant storage proteins found in the seeds of cereal grains. For example, gliadin in wheat and dynein in maize. They are characterized by their high proline and glutamine content, from which they get their name and are generally poor in essential amino acids such as lysine and tryptophan. And prolamins are predominantly present in wheat, barley, rye, and similar grains, and they can trigger reactions in individuals sensitive to certain immune system disorders such as celiac disease in humans. Consequently, individuals with gluten intolerance often tend to avoid food containing these prolamin proteins. And celiac disease, which is a gluten enteropathy, is a long-term autoimmune disorder primarily affecting the small intestine that occurs in people who are genetically predisposed. And I said it's a gluten enteropathy. In wheat, barley, and rye, prolamin proteins, such as gliadin, contribute to the formation of gluten along with gluten. This means prolamin proteins, anti-gluteins, prolamin proteins, and glutenins contribute to the formation of gluten, which gives dough its elasticity and the ability to rise during baking. The viscoelastic properties of gluten are crucial for the texture and structure of bread and other baked goods. And celiac disease is caused by a reaction to gluten, which are various proteins found in wheat, and in other grains such as barley. And classic symptoms include gastrointestinal problems such as chronic diarrhea, abdominal distension, malabsorption, loss of appetite, and among children, failure to grow normal. Proteins. Proteins are small, arginine-rich nuclear proteins that are typically associated with the packaging of DNA in sperm. They replace histones during the haploid phase of spermatogenesis and are essential for the condensation of sperm chromatin into a highly compacted structure. And this compaction is crucial for protecting the paternal genome during transmission to the oocyte. And human sperm DNA contains two types of proteins, PRM1 and PRM2, and both genes are encoded on chromosome 60p13. And we finish the globular protein. Second class of simple proteins, fibrillar proteins or fibrous proteins, the proteins whose molecule has the three-dimensional shape of a highly stretched ellipsoid. And scleroproteins. Scleroproteins are of animal origin. Scleroproteins refer to a class of structural proteins that contribute to the formation and maintenance of various tissues of animals. These proteins provide strength, stiffness, and structure to tissues such as skin, tendons, cartilage, and bone. They are resistant to enzymes such as pepsin and trypsin. And scleroproteins are characterized by their fibrous or fibrillar structure and usually form long, insoluble fibers. And the arrangement of these fibers contributes to the mechanical properties of the tissues that these proteins form. The unique properties of different scleroproteins contribute to the diverse functions and characteristics of various tissues in the body. And here are some examples: keratin, collagen, and elastin. And here you see the animation of collagen. And the collagen is the most abundant protein in the animal kingdom and an important component of connective tissue. It's an extracellular, inextensible fibrous protein that comprises the major protein component of such stress-bearing structures such as bones, tendons, and ligaments. It provides tensile strength tissue. And keratin, an important component of hair, nails, and the outer layer of skin. It hardens these structures and gives them strength. And keratin is very rich in sulfurous amino acid, cysteine, and methionine, especially cysteine. Elastin is a type of protein found throughout the body and acts mainly to maintain the elasticity of tissue. It is mostly found in connective tissue and is particularly found in the skin, blood vessels, lungs, and other organs that contain connective tissue. Elastin works together with the other protein collagen to maintain the strength and elasticity of tissue. Continue on fibrillary proteins, the fibrinogen, and here you see the molecular shape. It is found as a dissolved protein in the blood plasma. It is an acute phase reactant involved in the coagulation of blood. And during blood clotting or during coagulation, fibrinogen is converted to fibrin by thrombin. It is synthesized by hepatocyte in liver. Myosin, which is one of the three major classes of molecular motor proteins. The others are dynein and kinesin. And here the molecular shape, look at similarity in terms of being fibrillary or fibrous proteins. As the most abundant of these proteins, myosin plays a structural and enzymatic role in muscle contraction and intracellular motility. And myosin is a superfamily of proteins which bind actin, hydrolyze ATP, and transduce both. Most are located in muscle. And we finish this simple protein. It's time for conjugated proteins. Proteins which they contain other class of compounds, such as phosphate groups, carbohydrate groups, or metals, or nucleic acids or chromogenic matter. Let's start with phosphoproteins. And phosphoproteins are proteins carrying phosphoric acid as a prosthetic group. The prosthetic group phosphoric acid is attached to the hydroxyl groups of serine, threonine, and tyrosine in the protein. Or these are the amino acid residues carrying phosphate. And the examples are casein in milk, lipovitaline, phosphatin, and peptide. Glycoproteins. Proteins containing one or several oligosaccharide residues of different complexity covalent linked to a protein. And they are usually found on the outer surface of the plasma membrane, in the extracellular matrix, and in the blood. Some carrier proteins, such as ceruloplasmin, transferrin, and immunoglobulins from blood plasma protein, osteomucoprotein in bone, and also the other ones. We have too many examples which they carry carbohydrates as prosthetic group. Proteoglycans, they are macromolecules of the cell surface or extracellular matrix in which one or more sulfated glycosaminoglycan chains are covalently linked to a membrane protein or a secreted protein. So they are the proteins carrying glycosaminoglycan. But the glycoproteins are the proteins carrying oligosaccharide residues. And the glycosaminoglycan in a proteoglycan combine to extracellular proteins for electrostatic interactions between the protein and negatively charged sugar moieties on the proteoglycan. And they are the main components of all extracellular matter. The terms glycoprotein and proteoglycan refer to two different molecular structures formed by the combination of protein and carbohydrate components. Let us be very careful here. In glycoproteins, there is conjugation with the oligosaccharide chain, but in proteoglycan, with the glycosaminoglycan. And both play important roles in many biological processes at the cellular and tissue level. They are involved in functions ranging from intracellular communication to structural support of tissue. This figure illustrates glycoconjugates in mammalians. And glycans can be attached to proteins, as in glycoproteins and proteoglycan. And this is the glycoprotein moiety in a cell membrane, attached to the cell membrane protein, and this is a proteoglycan. And glycoproteins are decorated with N or O glycan. and N1 attached to asparagine, and the all glycans attached to serine or threonine residue of protein. And in proteoglycans, they are decorated with the glycosaminoglycans and mostly the sulfated ones. In here, the chondroitin sulfate and heparan sulfate. And also, don't forget, there are glycosapingolipids that we did in lipids. The glycan structures presented for each N-linked, O-linked glycosaminoglycan and glycosapingolipids also seen here exemplify one of the possible structures seen in cellular membrane. Yes, you see the lipoprotein. We have already elaborated in more detail on lipids. And lipoproteins are proteins or the conjugated proteins carrying lipids such as phospholipids, triglycerides, and cholesterol. They are easily soluble in water, and we studied chylomicron, VLDL, IDL, LDL, and HDL in lipid topic, but also there are other or different or variable lipoproteins. An example is here is the lipovitamin, a phosphoglycolipid. Look at the glycolipid, also this means also carbohydrate moiety, and it's a phosphoglycolipoprotein complex found in egg yolk. And it's a major protein component of the yolk and contributes to the emulsifying properties of egg yolk in food. Lipovitamin plays a crucial role in providing essential nutrients to the developing embryo in the egg and serves as a reservoir for lipids, vitamins, and minerals required for embryonic growth. This complex protein helps in the storage and transport of lipids, aiding in the nourishment of the developing embryo during the incubation period. Metalloprotein. Metalloproteins are biological molecular complexes formed by the combination of protein molecules and metal ions. These complexes often regulate the structural or functional properties of a protein. Metalloproteins play significant roles in many biological processes such as enzymatic catalysis, oxygen transport, electron transfer, and DNA binding. And here you see the rotating superoxide dismutase, which uses metals as cofactors, and there are different type of superoxide dismutases carrying zinc, iron, manganese, copper, and zinc. And it is most commonly found in eukaryotes that we have already taught in bioelements when we discuss zinc and copper. And here you see the human copper zinc superoxide dismutase with copper and zinc metal ions visible. And here you see the copper and zinc ions in the structure of superoxide dismutase. Chromoprotein. Chromoproteins refers to biological molecular complexes consisting of a combination of protein and colored components. And these colored components gives their unique features. And such proteins are often associated with pigments, which are responsible for the colorful structures of organisms. And colored proteins often have visual functions and form one aspect of biodiversity. And here, my friend, you see the example, the cytochrome C. It's a heme protein. Also, it's a metalloprotein because it contains iron. It is localized in the compartment between the inner and outer mitochondrial membrane, where it functions to transfer electrons between complex 3 and complex 4 of the respiratory chain. And here is the famous chromoprotein, as well as the metalloprotein, because it carries iron, as in the porphyrin complex, and then the heme, which make it also a colorful protein, a chromoprotein. And you see the porphyrin complex. There are 4 porphyrin complexes, and it's a tetramer, and also there are 4 iron molecules. And this is the space-filled model of hemoglobin. Each subunit, there are 4 subunits. Each subunit are given in different colors. Hemoglobin is an oxygen transport protein. Hemoglobin is an allosterate protein. It's a tetramer composed of 2 types of subunits, which are the alpha and beta subunits. And the 4 subunits of hemoglobin sit roughly at the corners of the tetrahedron, facing each other across a cavity at the center of the model. Each of the subunits contains a heme prosthetic group. The heme, the red ones are here are the heme group. And the heme molecules give hemoglobin its red color. Each individual heme molecule contains 1 iron atom. And in the lungs, where oxygen is abundant, an oxygen molecule binds to the ferrous iron. Don't forget, this is in ferrous iron. Ionic state is 2. And when the oxygen binds to the ferrous iron of the heme, then later released in tissues needing oxygen. The heme group binds oxygen while still attached to the hemoglobin monomer. The space-filled weave of the hemoglobin polypeptide subunit with an oxygenated heme group show how the oxygenated heme group is held within the polypeptide, as seen here. The red ones here. Anchoring of the heme is facilitated by the histidine nitrogen that binds to the iron. A second histidine is near to the bond oxygen, and the arms propanate groups of the heme are hydrophilic and face the surface of the protein, while the hydrophobic portions of the heme are buried among the hydrophobic amino acids of the protein. And perhaps the most well-known disease caused by a mutation in the hemoglobin protein is sickle cell anemia. It results from a mutation of the 6 residue in the beta hemoglobin monomer from glutamic acid to a valine, and this hemoglobin variant is termed as hemoglobin S. Nucleoproteins, another class of conjugated proteins. And nucleoproteins are conjugated proteins formed by the linkage of protamines, histones, and other simple proteins with nucleic acid. Nucleoproteins refers to biological molecular complexes consisting of a combination of nucleic acid, usually DNA or RNA, and the protein. And these complexes perform important biological functions such as storage, transport, and utilization of genetic material. Nucleoproteins can be found in the cell nucleus, in the cytoplasm, or in virus. And high nucleoproteins are found in ribosomes also, and cytoplasm, mitochondria, covalently linked RNA protein or DNA protein composition. And here were some examples. Histones and DNA. Look at the figure, histones and DNA. In eukaryotic cells, DNA often combines with histone protein to form a structure called chromatin. Here you see. And this complex contributes to the organization of genetic information and cell division. And we finish the structural classification of proteins. It's time for functional classification of proteins. In this class, we will start with the catalytic proteins and continue on transport proteins, nutritive and storage proteins, contractile proteins, structural defense, and at the end, physiological regulatory proteins. Catalytical proteins, these are the enzymes, are highly specialized proteins that catalyze biochemical reactions. And enzymes are defined as biocatalysts that allow biochemical reactions to occur rapidly under normal conditions and constitute the basic characteristics of living. And these are some examples: amylase, peptide, lipase. They are the catalytic proteins, or shortly, enzymes. Transport proteins. They are proteins that bind specific molecules or ions and transport them from one organ or tissue to another organ or tissue, or from one side of the cell membrane to the other. Serum albumin is the best known carrier protein. Bilirubin, calcium, fatty acids, and many drugs are transported by binding to serum albumin. Hemoglobin transport oxygen, lipoproteins transport lipids, and transferrin transport iron. Transport proteins found in the plasma membrane and intracellular membranes of all organisms bind glucose, amino acids, and other class of substances and transport them from one side of the membrane to the other. Nutritive and storage proteins that serve as biological reserve or reservoirs for metal ions and amino acids used by the organ. Albumin, main protein of egg white, and casein, main protein of milk, are nutritive proteins. They are amino acid stores. Many plant seeds also store nutritive proteins necessary for the growth of the germinating seed. The best known storage protein is wheat, and wheat is gluten, which is composed of gliadin and gluten. Keratin is the protein that stores iron. Contractile proteins. They are proteins that can contract or move spontaneously. Myosin and actin function in the contractile system of skeletal muscles and in many non-muscles. And myosin, together with dynein and kinesin, are the motor cells, are the motor proteins of the cells. And also there are other classes. And look at here, the microtubule consists of tubulin, and in dynein, and kinesin, and they deliver carbon. And tubulin, dynein, and kinesin are both crucial components involved in intracellular transport and cell structure within the cell. Tubulin is the basic building block of structures inside the cell called microtubule. Here you see the part of a microtubule. And microtubules form part of the cytoskeleton and perform a variety of important functions inside the cell. Tubulin is a heterodimer composed of two subunits, alpha and beta. And here we see different colors. And these heterodimers come together to form a kind of a long tubular microtubule. Microtubules are involved in processes such as mitosis and meiosis during cell division, maintaining cell shape, and transporting organelles within the cell. Tubulin also plays an important role in the formation of many microtubule-based cellular structures, especially appendages such as cilia and flagella. And these structures are essential for cell movement and various cellular processes. And besides cellular functions, tubulin is also important in specialized systems such as the nervous system. And dynein is a motor protein that moves along microtubules using energy derived from ATP hydrolase. It plays a significant role in intracellular transport by transporting various cellular components and organelles along microtubule tracks, which we call here cargo. And dynein typically moves toward the minus end of microtubules, and is involved in diverse cellular processes, such as the movement of vesicles, positioning of organelles, and the mitotic spindle during cell division. And kinesins are ATP-dependent motor proteins that can generate torque and displacement along microtubules, and they play key roles in mitosis and in the trafficking of organelles and vesicles. In summary, tubulin forms the microtubules that are essential for cellular structure and movement, with motor proteins dynein and kinesin facilitating the transport of cellular components and driving critical processes like cell division. And these elements exemplify the intricate machinery at work within the cell. Structure of proteins. The main structural tendons and cartilage is collagen, which has a very high tensile strength. Leather is almost pure collagen. Ligaments contain elastin and another structural protein capable of stretching in 2 dimensions. Hairs, nails, and feathers contain the other structural protein, keratin. And the main component of silk fibers and spider webs is different, another structure. The wing axes of some insects are made of resin. Defense proteins. They are proteins that defend organisms against invasion by other species and protect the organism from damage. Here, the famous ones are immunoglobulins. They are specialized proteins synthesized by the lymphocytes of vertebrates. Immunoglobulins can recognize and precipitate or neutralize foreign protein antigens such as bacteria, viruses, or other foreign proteins that invade the organ, in general antigen. Blood clotting proteins such as fibrinogen and thrombin prevent blood loss and the vascular system is injured by sealing the injured area with a blood clot. So the fibrinogen is an example of defense against blood loss. And the last member of functional classes of proteins, the physiological regulatory proteins. Proteins that assist in cellular regulation or physiological activity. Some hormones such as insulin and growth hormone are regulatory proteins. Insulin is involved in the regulation of sugar, carbohydrate, glucose, while growth hormone is involved in the regulation of growth. The cellular response to many hormones is mediated by a class of GTP binding proteins, often called G. Some regulatory proteins drop DNA and regulate the biosynthesis of enzymes and RNA molecules. And my dear friends, this is the end of the talk. Here are the references that you can check for further study. Next topic will be amino acids, peptides, and proteins-III, part 3 of the topic. And you can visit my website, and also you can follow my Instagram account for more about biochemistry, clinical biochemistry, and laboratory science. Don't forget to subscribe to the Biochemistry YouTube channel, like the video, and turn on notifications to be informed about news. See you in our next topic. Goodbye. Stay safe, my friends.